Human brain circuitry connects billions of neurons via trillions of synapses. Developing neurons project long branched extensions guided by extracellular cues toward specific targets to establish synaptic connections. These complex shape changes are led by a growth cone and require plasma membrane expansion and cytoskeletal remodeling. TRIM9 and TRIM67 are two E3 ubiquitin ligases required for shape changes in response to the extracellular morphogen netrid. Without TRIM9, growth cone have too many finger-like filopodia and axon branches and do not respond to netrin. Without TRIM67, neurons fail to send out filopodia and axon branches in response to netrin. Until now, the repertoire of molecular mechanisms by which these ubiquitin ligases work were poorly understood. Here, we used an unbiased approach to identify candidate interactors of TRIM proteins that modulate shape. A portion of the TRIM protein was attached to the enzyme BRA and introduced into developing neurons. BRA adds biotin to proteins near the TRIMs, like binding partners. The biotinylated proteins were clarified and identified by mass spectrometry. This identified a large group of interacting proteins, many of which were shared. This list includes cytoskeletal proteins and regulators, membrane remodeling proteins and regulators, components of numerous signaling pathways, and proteins that localize to pre- and postsynaptic regions. We validated one shared interactin partner, the unconventional myosin myo16, and found that myo16 was required for netrin-dependent increases in growth cone filopodia and axon branching. The large overlapping interactome of TRIM9 and TRIM67 likely contains additional regulators of neuronal form and function by which these two ligases coordinate neuronal shape and function.